Hi, welcome to the meeting. Doesn't look like anyone's here yet. So why don't we get started for this week? So if you're watching on YouTube, welcome, and hope that you find this information useful. This week we're going to go through a couple things. We're going to go through some open tasks. We're going to kind of review things as they stand and what we want to do for the rest of the year. And then the calendar year, that is. And then probably go over some papers and some things that we have in the queue. So let's start off here. Sorry. Let's start off talking about the major tasks. I will present my entire screen. And so this is the task board, or actually this is the task board. This is the task board for our group meetings task board. And so this has all of the things that we've been addressing throughout the year in our group meetings, things that are sort of loose ends or things maybe are done. We don't even have them in the right category. So that's why we go through this board. There's Jesse. Hello, Jesse.
Morning.
Morning. I'm currently going through the issues board. So why don't I go through the issues board, and I'm recording for YouTube. So and then we can talk about something afterwards. Oh, there's Krishna too.
Yeah.
Hi, Krishna.
Hi Bradley. Hi Jesse.
How are you?
I'm fine. How are you?
All right. We were just about to go through the issues board here.
Okay.
And then we can move on. Maybe did you have anything to present today, or?
Yeah, I'll present half of the research paper that we are writing, that reinforcement learning one.
Okay.
So first you can go on. I'll start in the end.
Yeah. And so this is the task board, and we haven't reviewed this in a couple weeks, but I think probably we can at least make some headway on it here. So why don't we look at what's finished here? That's always a good thing to do, to see where we are, to appreciate where we are. So we have the present collective pattern generators idea. So that actually was a neuromatch presentation. So this is done, but we still want to do things to follow up, and I'm not sure what those things are yet. I don't know if I think Jesse saw the presentation. I don't know if Krishna saw the presentation at Neuromatch, but the video should be up pretty soon. So. Okay. Anyways, yeah, so we want to follow up on this, but I'm not really sure how we would do that yet. We got to make some issues for that. And then we, of course, have this an associated paper with the bacillary and psychophysics and all that coming up early next year. We've already put the proposal in, so that'll be something to follow up on. Hacktoberfest is that kind of, I guess, fizzled out. It was just that we had a number of people who participated early, and then we left. We didn't get any more people participating, but I think we got about maybe 10 people to participate, which is actually pretty good. No one won any of the things that were offered. So, if you make more than 5 commits, you get a t-shirt. But, it's okay. It's just, I've not really done it before. We tried to do it like in 2019, but nothing ever happened. So we didn't get any contributors. So, so that's something that we can revisit next October, maybe. I'd do it again just simply because it's not that much you might have to do a little bit of advertising for the event for the month, but you can potentially get people to come into the platform that way. GSOC final presentations, that's really old. Create videos of diffusion limited aggregation. So I've actually created a couple examples of this and put them on the YouTube page. If you go to the YouTube page and look, just running some examples in NetLogo. I like the idea of making little tutorial videos. So what I did was I just ran a simulation on my computer. I recorded the video and then put it up on the YouTube channel as a sort of a supplement to some of the conversations we've had in the group here. So I don't know, I kind of like the idea of making little tutorials. I have some tutorials of other things up there that would be good to check out as well if you're interested. Yeah, this is the Neuromatch talk again. That goes back to the talk 1657, model of motile cells in the embryo. Not sure what that was. This lecture on PCA, UMAP, and TSNE, that was several weeks ago. So if you're, I think it was like 3 or 4 weeks ago. If you're interested in that, you can go check that video out. Or if you want to follow up on it, that's perfectly fine. Let's see. Revisions to the bacillary morphology paper, that's actually done. And all these are done. So I think we have a lot of, we've done a lot of stuff. So let's see, in progress, number 44, Krishna paper review. So that was the thing that Krishna is going to present on today. So that's going to be, I guess, finished, maybe. Well, we still have to review the paper, though, so it's probably not. But I'll look forward to that. This is the issue. Actually, I linked it to Hacktoberfest, tutorials for YouTube. So that's what I just mentioned, number 55. So if you're interested in making tutorials for YouTube on anything you can think of, from like simulations to maybe some other thing, we can talk about it, and we can put it on the YouTube channel and advertise it. The DevoWorm bibliography. Actually, I've been thinking about this more, and we've talked about bibliographies as like putting it in something like EndNote and having a list of references. But I actually found some interesting templates for, and just as an aside here, and something called periodicals for annotated bibliographies. So if you've never heard of annotated bibliography, you'll see that it's basically, a list of references, and then there's like a description underneath the reference, and then from those descriptions you can sort them out. So like this one is The Life of Behavior. The authors are here. This is the date of publication, the DOI. And then this is actually when it's been posted, October 14th, 2019. In this paper, the authors remind us that neuroscience or objective studies living stuff. And so they go on about the 3 essential principles of behavior. And so you can have like a reference, its identifier, and then this blurb underneath that tells you what it is. And then you can have, you can link those different entries in different ways based on the description. So this is actually paramecium neuroscience. So this is the neuroscience of a unicellular eukaryote. So this is familiar because we've been talking about bacillaria and neuroscience. But it did the same thing here, where they have these descriptions of the reference, and kind of like some background. And so that might be an interesting thing to build for some set of topics. I did this when I was an undergraduate. I built a annotated bibliography, and they're quite useful. I think that's something we could maybe do as well. So that's part of this bibliography thing. We have a couple issues outstanding on that, but that's something we can update later. Create an outline for action items on periodicity paper. So last week, I think Susan was in the meeting and we talked about the periodicity paper, which I haven't really made that much progress on it, but it's where we have a Google Docs draft of this, and maybe next week I'll go over it a little bit more, talking about like where we are. I'll try to make some more progress on it and try to figure out where we are and how people can make contributions to it. I know we'd been talking about it earlier this fall, but things have come up and we've pushed it back, and I think it's probably time that we revisit it, because I think there's a version of it due to a special issue that we want to target at the end of the year. So axolotl embryo animations and segmentation. I think this is something that Ojwal was working on, but he's been not feeling well the last couple of weeks, so he hasn't been coming around the meetings. But this is something we want to keep on the sort of the mid-range burner, because we, it's an interesting area. We have the data, and we're, it would be a nice addition to what we have in the group. Let's see. Oh, we've got a new member here. Hello, how are you?
Bradley?
Yeah.
Yeah, she's from my college. When I was advertising for the Hacktuber, she was interested in, contributing to our open source organization.
Okay.
Yeah, I know her. She's from my college.
Okay. Hello, how are you?
Yeah. And
Hello, sir. I'm good, sir. How are you, sir?
all right. So you're from Krishna's college, the same college?
Yes, sir.
Okay. Well, welcome.
Thank you so much.
Yeah, I guess Krishna will be presenting in a little bit. And currently I'm going over our task board, so this is actually a good open source experience here.
Actually, yeah, that's what I'm new to open source, so I don't know much about it. I was very interested, and then I saw Krishna's status, and I asked about your organization, and I thought maybe I can join in and learn something.
Okay. Well, it sounds good. Yeah. So, yeah, welcome. And, so we were talking about this thing called a task board. And so this is where we put a lot of our tasks. And we use GitHub here for our task board. We generate tasks in GitHub, and then we revisit them. We put labels on them, perhaps, and then we review them every once in a while in our meetings, maybe every couple weeks. So this time we're going through, so we go from like right to left. We go through finished, in progress, and then to do. And then, it kind of helps us think about like what we're doing in our meetings. So if we do something, a lot of things we do in our meetings, they just kind of maybe would otherwise fall through the cracks. And we're trying to, keep on top of a lot of different things. So that's one thing about like an open source organization, is that we like to review these progress or task boards. And so this is something that's publicly available, so people can view it. They don't have to register for anything to look at it. But there are a bunch of tasks on here that anyone can participate in. So we have a open paper here, periodicity in the embryo, that we're working on, for example, which is a thing that if someone's interested in participating in, they can inquire or look at the paper on. I don't know if I have a link to the open paper, but basically, people can contribute to these issues as they wish, if they have something to add. And that's the idea here. So we have a bunch of ongoing projects. One of those is complexity measures, number 3. So complexity measures, I don't think we've kept up on that too much. We've talked a little bit about different things, but we haven't really gotten any momentum on that. that's something that I think Jesse was involved with, Dick was involved with. So we'll probably update on that soon.
You may have stopped sharing your screen, by the way.
Oh, okay. Let me reshare it. Maybe sometimes that doesn't work. All right.
Happens to be me.
I can reshare it. Sometimes if I'm flipping between tabs, it'll... Okay. Now do you see it? Okay. So this is...
I don't see it, but I don't know if other people do. It might be my device.
Oh.
Oh, okay, yeah.
Okay.
For some reason, you were pinned on my device, but I do see it now that I'm unpinned you. So, yeah.
All right. Yeah, so the complexity measures is open. We've had prior conversations about different things like, different network measures and different... we have this collection of materials. So recapping, we've talked about trees and metabolic scaling and information measures. So there's a lot of stuff there. But I think that's probably something that if, people have a good amount of momentum on it, they can start to move on it if they want. I think I lost my thing here. Why did I lose it?
Yeah, I'm obviously I'm going to be busy for a few weeks in particular with different stuff, but when I come back, I would like to look on. I sort of was a very, very limitedly pushing forward on that front, but I think I'll return to that once I have more free space to do it.
Yeah.
That's in that project for sure.
Yeah. So, and then this bacillary non-neuronal cognition paper. So this is the thing that is linked to a lot of these issues with the presentation at Neuromatch. And so...
Is that... I feel like I keep asking this, and I apologize for that. Is that paper actually done, or are we still working on that paper? Could I join that paper? Is that done?
Oh yeah, you can join it. We're still working on it. It's sort of in a state where I think we have a good main idea, but we need to work on some of the fleshing out the details of the measures and how they apply, and it's still not clear whether we can actually do a formal analysis. But we can at least propose some measures and things like that. So I think we'll probably follow up with what was in the presentation for at Neuromatch, and so that's something we can work on the next couple weeks. There's a deadline on that of early next year. So I submitted a proposal and it was accepted. So I think it's March or something. So we've got a little bit of time on that. But yeah, it's still very much not done.
Okay.
So, and then we have some more issues in hold on axolotl data analysis. So this is Susan's data set that she's opened up for us, and we're thinking about ways to analyze it. And I know, again, Ujjwal had some ideas, but he's been out of action for a bit. So we might revisit that soon. But we've still got that on top of our minds. So Krishna proposed last summer a general biological ML model, and so that's also something that is one of those big ideas that we have to kind of get our hands around. I know this is a little, this is a little bit of a reach for a quick project, but have you thought, Krishna, you've got that paper that you're working on, so maybe that is tied into this as well, right?
Yeah.
Okay. Then there's this create embryo model in Blender number 38. So that's, again, something I think Ujjwal was working on. I don't know what the status of that is, but we'll find out maybe in next couple weeks. He was working on a 3D model of embryos. So Blender is a 3D modeling program that you can render 3D objects in. So he was taking data from imaging data and turning it in, extruding the objects and turning them into 3D objects. So, and we're still trying to get something for the container, for the Docker container for OpenWorm. So that was kind of the idea there, that would go into that container. But I don't know if we'll have to see how it develops over time to see if that's suitable. Recruit people as DevoWorm contributors. So that was tied to the Hacktoberfest initiative. And I don't know if we need to do a post-mortem on the Hacktoberfest stuff. But like I said, we had about maybe about 10 people participate. Maybe a couple people participated multiple times, but not enough to get any sort of prize from GitHub. So, the idea was if you did 5 pull requests, you'd get a t-shirt. I think we got 3 was the top number of pull requests. And so, that people didn't get t-shirts, but I liked the interaction on DevoWorm. It was sort of all bunched up at the beginning of the month, but, I think people forget about Hacktoberfest after a while. I didn't really advertise it after maybe about the 6th of the month, so, just kind of got busy. And
I don't know if you get to see as a repo owner, but I think you can. I think it's that you can do a bunch of pull requests, but they don't have to all be in the same organization. So maybe they did get T-shirts, just not.
Yeah, a total of 5 requests are necessary. You can even create a pull request on your own repository. You just have to give it a label of Hacktoberfest.
Oh yeah, you do have to label it. That was one thing we didn't actually do at first, and then about a couple days in we figured it out. I don't know if you're involved in that, Krishna, but maybe. But we got that. We labeled the different so most of the repositories in DevoWorm are labeled Hacktoberfest. But I don't know if you can go between organizations too. Like, I don't know if anyone committed in other organizations and then ended up getting their prize, but I like the idea of Hacktoberfest, kind of being invested in it a little bit. I don't know what we could give on our end in terms of like prizes. That would just stimulate people to contribute more, like, some sort of like recognition, or I don't know if we have any swag that we could give them, but that's also something we could do outside of like what GitHub's doing. Like, if you make maybe like 2 commits or a really good commit, you get some prize. So, I think I did recognize everyone who made a commit, like in the newsletter. So I put their names in the newsletter. And, but yeah, I think if we do this next year, it'll be interesting to see if we can get more people. How to promote it now a little bit. So, we'll look into that next year. But I think it went pretty decently this year. I've never done it before. And some orgs are pretty big and they have a lot of promotion. They promote it very heavily. So, then there's this embryo visualization task number 6. That's for the OpenWorm Docker. So that's what I was talking about when I talked about the embryo model in Blender. That's connected. So OpenWorm Foundation has this Docker container, which is it's a virtual machine that you can download to your computer, run it, and it's supposed to give you the full OpenWorm experience. So every project in OpenWorm has a program that opens up and runs, like a simulation of the worm, or like a model of the worm. There are all these different applications for each project, and DevoWorm does not have an application. And the reason for that is because we really haven't been focused on just easy to execute simulation. We've done a lot of data analysis, we've done a lot of machine learning, we've done a lot of theory and other things, but not really anything as straightforward as what some of the other projects have done. So we've been thinking about how to build a Docker showcase app or something. And so this embryo model might be one example, but it's got to be something lightweight. It can't be something really, because we already have a lot of apps in that Docker container that are, generate a lot of output data or high memory applications. So we're still searching for that thing to put in there. But I thought I'd bring that up because we keep mentioning it, and there's a lot of interest, but then it's hard to really know what to actually put in there. And that's no one's fault. It's just it's a hard problem to kind of do. So we have a couple of these open papers, which are basically papers that we have that anyone can contribute to. I think I need to make that more prominent on the website because I've talked about this in meetings, and probably isn't clear to people where to go for that. I don't know if I have a link to it in this board, but let me see. Yeah, I don't think so. Well, anyways, we have a spot on the GitHub where you can put, I think actually it's in group meetings, if I'm not mistaken. Maybe not. Well, anyways, let's see. Yeah, group papers. So we have these group papers here in the README of group meetings. And there are a couple of open papers. So some of these, one of the ones I was talking about was this bacillary non-neuronal cognition sub-project. And that has 2 open papers. And so they can either be Google Docs or a GitHub repo. So this paper, say like emergent behavior in C. elegans and other organisms. If you click on the GitHub link, you come up into this, and it's not really a paper, it's an outline at this point. But this is the idea here. If you read through this, you'll see where this is going. And then if you're interested in contributing, you issue a pull request, which is where you can fork the repository and then work on the thing and then issue a pull request, and then you can have your work accepted. Or you can just edit it in the document. So here, if you wanted to edit this document, you'd hit this pen, and you'd have to create GitHub will create a fork for you, and then you would just issue a pull request on that fork, and then push it up to the main directory. And so this is this open papers idea. So this is a way to get people involved in writing papers who might not otherwise have access to it. Some of these are in Google Docs. So if that's the case, you go to the Google Doc and you request permission, and then I can give you permission, and then the same principle applies. You would make your changes and you make a note of what the changes were. It's a little bit different than GitHub, but so that's if people are interested in participating in open papers, that's a good way to go. So then finally, I think we have, let's see, any more issues? Oh, the create a theory layer for DevoWorm. So the DevoWorm organization have created a couple new repositories there. One of them is called Theory Building. So this is the idea of creating a theory layer for DevoWorm. So we have the machine learning resources, we have the data science resources, and we also have this theory building layer, which is sort of in very early days. I need to sketch it out more and put some conditions on what we're talking about there so that people don't come in and they're just kind of lost. Maybe we'll take some of the ideas that we've been working on, some of these other projects, and make them sort of into principles for contributing. So, we have this data analysis that we've done on a lot of these things. What is what's the theory behind what we're doing when we analyze the data? And so that's why I have this theory building layer. And so I think we'll move on now from that board, and we'll be reviewing that again in a couple weeks, to the DevoWorm repository. So this is the current state of this. We have an education repository. We have the data science demos repository, which are a series of data science demos that people have created. We have DevoWorm organisms. So these will be like different model organisms, and we'll put in resources into these folders as they come up. We also have the community board, which is basically the project board for DevoWorm. And then we have finally this theory building repository, which is basically the theory behind a lot of the things that we're analyzing. And so that's the way we're going to have this. Under education, I wanted to bring this up. I've been working on this Journal of Open Source Science, or open source, whatever it is. So anyways, this is a publication that builds upon last summer's GSOC projects. So I've gotten it actually into the form that they wanted, I think. Pretty close. I think it's Mayoko who created this little template. And so that's what this is here. It's a little table that has like the title and the authors. So it's Mayoko, Ojwal, myself. And the summary of this, so if you're interested in looking at this and reviewing, maybe you can provide some comments if you wish. Where's our chat window? Here it is. So this is a very short this is a journal that's run entirely on GitHub. And the idea is to give people who make open source software a place to publish a short descriptive paper. And so this is supposed to be about maybe 1 to 2 pages in length. And so we have the summary section, which summarizes what it is, links to it, the repositories, then a statement of need, which is, that we have this organization, we have this need to take data and quantify it and explore it a bit with, machine learning tools, deep learning tools. And then we have this, these are some schematics showing the DevoWorm platform, the DevoWorm program, and then how to execute this. And then we have some references. So this is I'm going to add a couple more things here. I need to add a section on, I think I already did actually add this section on meta features. But there are a couple more things I need to add to it, but we're making progress on this. And I thought I'd bring this up because I've been putting this off for a long time. So that's all I have to talk about the world of our GitHub and updates. Now, Krishna, you had a presentation you wanted to make on your paper.
Yeah, am I being visible here?
Oh, yeah.
Yeah. So, first, this is the paper that we are planning to, it's not completed. It's on reinforcement learning. Is my screen visible?
Yes.
Yeah. So, it's on reinforcement learning. If I have to explain it in simple words, it generally works on reward policy, reward and punishments. For example, if you are having an agent and we want it to do a certain thing, like for example, if we are having a drone and we want it to learn how to fly, so when it crashes, we give it a punishment, or you can say negative of a reward, and when it flies in a good manner without crashing, we give it a positive reward. So here, the main focus of the agent is to maximize its points. For example, a person is playing chess, and if we have that, if you hit the pawn, you will get that much of points. If you hit the rook, you will get that much. If you go for the knight, you will get that much. So at the end, the person has to maximize its points. So same goes with the reinforcement learning. So SARSA is, you can, in simple words, you can say it's a method for Markov decision process. Uh, 1 minute. Yeah, for example, if we have to do a certain number of tasks, and we have like 1, 2, 3, 4, 5, 6 here, it's a Markov reward process of a robot. So if it does task 5, it gets a reward of -1. But when it's shut down, it doesn't get any reward. Whereas if it does task 4, it gets a positive reward. So you can visualize it that task 1, 2, and 3 are crashing to a wall, and task 4 is going successful to its end point. And refuel is just the way when it fills it petrol. So for example, if we have that red dot if starting point, and we have to reach to the star, and all these purple blocks are having -1 reward. So what we have to do, that we have to reach to the star with the shortest distance possible. But if we complex the program and we add hills between, then now our agent has to go to the star, but it has to avoid the hills because they are having -2 reward, because they are difficult to climb. So here we can have a number of ways to go. We can follow this thing, we can follow this route, or we can follow this route. So after all the routes, our agent learns that whenever it goes through the hills, the reward it's getting is the least one. So it try to maximize its chance not to visit the hill. So there are two types of reinforcement learning thing. One is on policy and one is off policy. If you are going on off policy, if we have 3 routes, route number 1, 2, and 3, and when we go on the route 3, we get a negative reward. So we come to know that our way is on route number 1 or 2, and when we go on route number 2, we again get a negative reward. So we come to know that our route is 1. So here you can say we are excluding the choices, and we are deciding the route we have to take by the other routes. But in case of on policy, that is SARSA policy, we go to route number 1, and then we see, is it feasible? So so this much work has done. It's regarding I have done about the abstract. I have explained what are Markov reward processes. I've given few examples, and it's rough about 5 pages, and I have to add you can say bibliography. I have to add references, and done a comparison that's not completed yet. So this is the first paper. Anyone wants to ask any question or give any suggestions?
Do you have anyone have any comments before I comment? I mean, I think it's a good paper. I don't know, like, I've read a little bit of it. Of course, it's incomplete, so it's hard to say. Are you planning on doing like benchmarks or examples?
Yeah, it would be much more of a literary survey.
Okay.
It wouldn't. I can do the benchmark scores also, but I'm thinking to present it into much more of a theoretical way. If I go into the benchmark also, it would be become a lengthy one, so it would take even more times. It's being incomplete from you can say past 1 month, and if I have to add the benchmarks, it would take another 1.5 months.
Right.
So, yeah.
This idea of off policy and on policy. So this is like, it almost sounds like off policy is like exploration versus
So we also have a thing. It is exploration versus exploitation. For example, if we are having 10 routes, and from first 3 routes, I come to know that the first route is the better one. So there is a dilemma of exploiting the other routes or you can say going with your existing knowledge. So if I'm able to explain it. Hello.
Yeah.
It's a dilemma between exploitation and exploration. That either I want to exploit all the possible routes, or with my existing knowledge or with my existing experience, I want to stick to that and make the reward as much as I can. Because if I go on exploring, there are chances of getting even a better path or even getting a worse one. So here the dilemma comes into play. So I'm going to go into detail of that in the, you can say, later section of the paper.
Yeah. That's an interesting area to explore, I think, too.
Because it's a dilemma kind of thing that you want to get out of your comfort zone, or sometimes getting out of comfort zone can not be much useful. So here I'll explain that in the latest section of the paper. And now I have to present the second thing, that general biological CNNs.
Okay.
I discussed. So, one minute. Yeah.
Okay.
Is everything visible?
Yeah.
So, one second. Yeah.
Okay. Okay.
So my proposed project is a custom-based convolution neural network for biological data. As we people know that most of the CNNs that we are using are trained on ImageNet dataset. It's a dataset that comprises of vehicles, traffic lights, cup plates, human beings, cats, and dogs. So all the state of the art CNNs, for example, the ResNet, the LXNet, all of them are trained on that dataset. But then they are retrained on the area of interest dataset. For example, if I want to classify benign and malignant cancer, first I'll use my ResNet that has been pre-trained on the ImageNet, that dataset that contains cat, dogs, cup plates, human beings, and vehicles, and then I retrain it on my own data. But I guess this thing can be improved a little bit. It's a tedious task and you can say a big project. But if we take all the biological data and we pre-trained our network on the biological data instead of that cat, dog, animal, human beings, and vehicles, and then we retrained it whenever a person wants it to do. For example, when a person can use ResNet in any way, but it is pre-trained on a broader spectrum. But if we narrow down our approach and we pre-trained the ResNet on the biological data, and then if a person can retrain it according to his own whims. So convolution neural network, where we take an image and you can assign its importance. That is just the learning weights and biases. And so why pre-trained data? Because if you want to train something from scratch, it would take a lot of time and it would consume a lot of computation power. It would not be feasible with time and with economy. So what we do instead of that learnable weights that I discussed in the previous slides, that they are not initialized from zero. They have some random value that was learned during its initial training on dog, cats, human, and vehicles. So once if we are not starting it from zero, they tend to perform better, and they tend to learn fast. For example, if a person knows how to drive a car, so he'll be able to drive a bus or some big vehicle in a better way. But if we are teaching a person to drive a 16 tired bus or a big Chevy truck, so he's more prone to crash. So here, what we are doing, we are not initializing the weight from zeros. Our model knows something about images, but not particularly the types of image that we are going to feed. But if we narrow this approach just for biological data, and we have plenty of it, for example, if for example, if we want to predict cancer, and our training data set had cancer images, plant images, zygote images, microbial images, the probability, or you can say the what you can say, they all these type of images will share a common thing to some extent. A cancer cell would be distant to a vehicle than to a plant cell. So even though they are varying, the plant cell and animal cell varies a lot, but at least they don't vary as much as a car and then a cell. So what are the benefits of pre-trained model? So it's very simple to incorporate. Once we code it, and we put all the weights and biases, and create a model, then any person with not even much knowledge of deep learning just has to import the model. You can say give his training data and get the outputs. So even a person with little bit computation knowledge can perform it. The second thing is that there is, as mentioned earlier, the results are they are quick to get, and since there is not much label data, so if I have pre-trained my model on 1,000 images, then it would be able to perform even on 100 to 150 images when retrained. And the second thing is versatility. For example, if I am taking the broader sense of biological data that includes plant, animal, and all the things from microbes to a cancer cell, from an embryo to everything possible, whatever label data that we can find. So we will have a lot of versatility that even a botany person dealing with plants can use it, and you can say, for example, a person working on animal data can also use it. So we will have a lot of versatility, even though that versatility is less as compared to a normal ResNet. Yeah, but it would be, you can say, focused on biological data, and it would be versatile on every biology domain. So yeah, first of all, it would be also have a few of hurdles. First is computation power. If we want to train 1,000 of images, we have to train them from scratch. First, we have to collect all of the biological data that we can publicly get. And since biological data, especially concerning with human beings, is difficult to get because of the privacy concerns, but we do have a few things like Radiology Association of Northern America, RSNA. We have a lot of public data, but it will be a tedious task to accommodate it. It would take a few weeks, or you can say 1-2 months. But yeah, we can do it. We have a lot of things like Kaggle and Google Open Data Sets. But yeah, we have to collect it. Then we would be needing GPUs for training it. That would be also, you can say, it is difficult, as the free Google Colab that we get has limitations. It can be only done for certain hours in a month, and its GPUs are like K80. They are not as good as the V100 or P100. The amount that will the amount of time that would be that we would spend on Google Colab would be 1/10th of the time that would be actually we will need if we use a better GPU like RTX 3080 or V100 or P100. So that would be another limitation. But once we have successfully applied the pre-trained model, I guess we would be uh, first one, first one of the open source organization that would have just focus the biological data in case of CNNs. We do have UNet, that is a segmentation type of model that is used in biomedical analysis. But we don't have a journal biological model that would be used in biomedical to an normal animal to plant data. And that is what I earlier explained, that image net data exclude these type of things, and our biological data is this. So I guess there is not even 0.1% of similarity between them. And that's just the thing that I earlier mentioned. Yeah. Any questions? And this is the second time I'm presenting this.
Yeah, I think it's good to have a refresher. Glad you've been thinking about it. Does anyone have any comments or questions about it?
I'd just say, excuse me. I'd just say, I really like the presentation. It was, I feel like I learned things from it, and it was well explained. So I think it was a very good presentation.
Yeah, thank you. I'll send you the link on Slack. I'll share the presentation on DevoWorm channel.
Okay. Shruti or Susan, do you have anything else to add? Any questions or comments?
There should be a way of getting some medical data from data sets. Like sometimes you can get open source ones. I'm not sure about it, but I know there are medical data.
There is plenty of data. We just have to collect it, and it would be it would be a manual task. But there exists a lot of data, especially in case of cancer and radiology field. We have a lot of data. The issue would be in plant-related data, but that would be also in abundance if we try hard and spend some time on collecting it. And we would be, I guess, the first one. I'm not sure that someone is working on specialized biomodels. People are working on biomedical models for disease prediction, but in general, no one is working on bio data as far as my knowledge is concerned.
Okay, I'm hoping to generate some more data, more biological data, using optical coherence tomography, and apparently you can use it for histology as well. So that might be another type of data set.
Yeah, that would be great, and that would be a rare form of data that is can really give versatility to a model.
I think that's great. I like the focus on pre-trained models. So, this last summer we did a pre-trained model, but I think it's a very rich field. I'm not sure. I think it's absolutely necessary for if you're dealing with a lot of the biological phenomena that we're interested in this group. So, you can do. I've seen actually people have built pre-trained models for different features of a single cell. So there's some pre-trained models that exist for segmenting mitochondria out of cells and things like that, or different organelles. And then there are, of course, pre-trained models for cells. But then you have broader features, which we've kind of touched on in the DevoLearn platform, where you have motion cues and things like that, things going on in the embryo that exist. So they're sort of at the sort of multicellular level. So, and especially something I know you've seen bacillary in this group, but more generally with a lot of multicellular colonies, you see different features that are either emergent or they're sort of collective behaviors that you want to capture, and so those are, you need to have a pre-trained model for that because it's hard to label those events. They're just things that kind of emerge out of the soup. I know we've talked about, like chemical reactions in this group before as an example of collective behavior. Well, those sorts of things are actually a pretty good example of something that you can't really label very easily, but you nevertheless are interesting features. And so, but you have cells, you want to know the features that are single cells, but you also want to know the feature which is the collective behavior of the cells. And that's where it gets tricky, because you could probably do you could probably pick out features that represent sort of collective states of matter, like some reaction where you have mixing or something like that. But, to have that sort of two-layered thing where you're looking at cells and then you're looking at this collective thing that they do. I think that's actually quite interesting. So, I like the idea of a general biological model. I think the way I would go forward on this is maybe thinking more about what are the types of features that might span a lot of different areas of biology. So are there features that are, common to all different types of cells? Are there features that are kind of common to what cells do in collectively moving around or configuring themselves? Because then, you have things that you see either in medical images or in
Actually, our group is having people with different domain experts, so we can leverage that. We have people with biology and computation, and it's a mixture of both. So we can learn from each other's domain expert, and you can say that make it state of the art. We have that leverage of that thing.
Yeah. Okay. Now I know it's the top of the hour, but I did want to get to the papers some of the papers that I had. I'll probably be about 15 to 20 minutes on this. So if you have to leave, that's fine, but I'm going to go through these, and this will be on YouTube later. So thank you, Krishna, for your presentation. And let me... What was that?
Nothing.
Oh, okay. That's fine. So let's see. I'm going to present here my entire screen. So I just wanted to go through some papers that I find interesting here. I talked about these two earlier. These are annotated bibliographies. And if you missed this part of the conversation, it's probably about 10 minutes in. You can watch it on YouTube later. But it's basically this idea of building these annotated bibliographies, which have each entry is a paper, and then you have a short description underneath. And then it could be the author or the reader's reflections, or it could be just what it's about. But then you can actually link these together in a search or some other way. And so that's potentially useful for the group. But that's these two. But I want to talk about some of the other things that I found in the literature here. So this is a paper that this is something actually that is relevant to both. So I have another group on Saturdays. We talk about a lot of things on AI and developmental AI and things like that. And so we've been having these conversations. We actually did a presentation at Neuromatch on developmental AI. And I found this paper yesterday, and so it's more of a neurobiological paper, developmental neurobiology. But I think it's really interesting, and I'm hoping that we can maybe incorporate it into that work. So this is called Fate and Freedom in Developing Neocortical Circuits. So it's not like C. elegans where you have a set of cells that are connected through a connectome. These are human brains or mammalian brains where you have a neocortex, and then that neocortex is densely connected, and there's, the basic idea, we'll go through the abstract, and I'll give you the basic idea here. So the activity of neuronal circuits of the neocortex, which is the front of our human brain, which is the enlarged area, and you all know what that is, but and of course this exists in all mammals, underlies our ability to perceive the world and interact with our environment. During development, these circuits emerge from dynamic interactions between cell-intrinsic, genetically determined programs and input activity-dependent signals, which together shape these circuits into adulthood. Now, this sentence here is interesting because I've been thinking about this in terms of when we talk about we experience things in the world versus the sort of the genetic underpinnings of neurons and how they develop. And so I guess if you're going to model this actually requires two things. One is that you have a neuron or something that represents a neuron, but also something that represents the genotype. And then you have some way of characterizing environmental information. And so my other group were talking about these issues, and they're not quite gelled yet, but I think this is a good sort of framework for thinking about this. And I'm going to propose this in the Saturday group next week a little bit more formally, but I'm just previewing it here. I think this is relevant to DevoWorm because we need to talk a lot about cells and genomes, but then also maybe what cells are doing in response to that. So building on a large body of experimental work, several recent technological developments now allow us to interrogate these nature-nurture interactions with single gene, single input, single cell resolution. And so here they focus on excitatory glutamatergic neurons, which is a class of neuron defined by its receptor type. And this review discusses the genetic and input-dependent mechanisms controlling how individual cortical neurons differentiate into specialized cells to assemble a stereotypical local circuit with global large-scale networks. So these local circuits are being integrated into networks. Cells are differentiating, and he's talking here about sort of the way we can measure this. And so this is an interesting paper. They kind of go through this idea of cortical columns, which, if you're familiar with mammalian neocortex, this is the way these cells are organized into these layers. And so it's a very interesting paper for a lot of reasons. Kind of walks through a lot of cortical information flow, direct and indirect neurogenetic pathways during corticogenesis. So cortex has its own method of neurogenesis. So cells are born and they move through different layers of the cortex. So they move to their final position, but there's a whole process of neurogenesis there. And it involves significant migration of the cells. What else? This is self-organizing properties of neocortical neurons. So it kind of gets into this idea that neocortical circuits are self-organized. So we have this model here of different cells in different layers, and they start to connect. And you get this sort of relationship between the cells and how they form their connections. So this is a good paper if you want to read more about it. I found this really fascinating. The same author, along with some co-authors, also wrote this paper, Cross-Modal Genetic Framework for the Development and Plasticity of Sensory Pathways. And so this is very similar. It's a little bit more data-driven, I guess. So this is, again, the neocortex in mammals. For each sensory modality, input follows a cortico-thalamocortical loop in which a first-order exteroceptive thalamic nucleus sends peripheral input to the primary sensory cortex, which projects back to a higher-order thalamic nucleus that targets a secondary sensory cortex. Basically, what he's talking about is that you have this structure called the thalamus that sends projections up to the cortex, the thalamus taking in connections from other parts of the body, and then it sort of loops between the thalamus and the cortex multiple times. It sort of is a sampling of information. And so this is another interesting paper. It's pretty dense and technical, but I think if you are interested in that topic, it kind of opens your eyes to some things. Another paper, and this is quite a bit off in the other direction, is called How Do Living Systems Create Meaning? So this is by Chris Fields, who's an independent researcher, has published a lot of interesting papers on like philosophy of science and other things. And then Michael Levin, who's a developmental biologist, computational biologist. And so this is quite a bit far away from what I was just talking about, but it's interesting because we're also talking about in this group things like basilaria, that we're talking about information processing and cognition. And so Michael Levin is also interested in sort of non-neuronal cognition, and they've been talking about this problem. And so they've kind of come up with this idea of living systems creating meaning. And so there's this earlier field called biosemiotics, which kind of dealt with that a little bit, but they're kind of taking another stab at this. So the abstract reads, meaning has traditionally been regarded as a problem for philosophers and psychologists. Advances in cognitive science since the early 1960s, however, broadened discussions of meaning, or more technically the semantics of perceptions, representations, and actions into biology and computer science. Here review the notion of meaning as it applies to living systems, and argue that the question of how living systems create meaning unifies the biological and cognitive sciences across both organizational and temporal scales. So they kind of get into the idea of meaning in biology as opposed to like in brains. Well, we know that like brains generate agency, and so people propose, well, cells do this too, they just don't have brains. And so then the question is, are we making are we projecting ourselves onto cells, or is there actually a mechanism for this? And so when you ask this question, this brings up a bunch of things that are sort of related. And one of these is this idea of meaning. And this is related to agency in terms of is a system that doesn't have a brain, can they generate this kind of, meaning of things? This sort of semantic layer. So that's the idea here. And so, let's see, is there anything particularly interesting down here? So, in contrast to this focus on individuals and learning, we advance in this paper a deeply evolutionary approach to meanings and suggest, consonant with the theme of this special issue, there's a special issue involved, that the construction of meanings present a common and pressing question for all disciplines in the life sciences. We focus in particular on three questions that are foundational to the study of meaning. So, the first is, how do living systems distinguish between components of their environments, considering some to be objects worthy of attention and others to be background that is safely ignored? That doesn't necessarily mean that there has to be a meaning system or a semantic system. It's just one of the things that organisms do, and maybe even single cells do. It's like a signal detection thing where you're looking at signal versus noise. It's like, are you looking at some object, or are is there something you can identify, like a chemical signal, or some object like another cell, or is it just background? Is it something that you don't have to pay attention to? In humans, we use something called attention, which is a really complex system in the brain, but you can do this with a simple signal noise detection sort of mechanism. But that's one question. But the second one then is, how do living systems switch their attentional focus from one object to another? Well, now they're using the language of attention. So this is what this is signal versus noise. You're rejecting the noise and accepting the signal. Now you assume that attentional drive is focused towards a signal, hopefully. And so now how do you switch your attentional focus to different signals? And then the third question is, how do living systems create and maintain memories of past events including past perceptions and actions. Now they're proposing a memory mechanism. And in that case, we know that single cells actually do have memory mechanisms, like epigenetic mechanisms, or in one of the things that we proposed in the Basilaria presentation, there are different extracellular mechanisms, like polymers that can be used as markers, as like memory devices outside of the cell, so that the cell, has like something that it can draw upon to guide its behavior. So you might have pheromone trails, or you might have chemical gradients that you follow. Those are all memory systems in a sense. Then these questions all presuppose an answer to a fourth question: how do living systems reference their perceptions, actions, and memories to themselves? So that's actually important, because all of these things that we're proposing, they're kind of tenuous in terms of how we think of them, but they are like they're actual mechanisms in the cellular world. But then how do these things get referenced by the cell? Like if there are things out in the environment that exist, like a chemical trail, and the organism or cell follows that chemical trail, then how does it reference it? Like, how does it know where it is in the chemical trail? How does it know when to stop? Is it just a bunch of signals that it's just kind of deterministically, responding to, or is there some sort of system of meaning and reference, sub-referencing that goes on in the cell or the organism? So they say we use the term living system instead of organism in formulating these questions to emphasize their generality. So again, if we're dealing with anything from like complex organisms to single cells, we have to be flexible. And so this paper kind of goes through this, and they kind of consider maybe a meaning system within a single cell or within a living system. So they're very general about it. And so they're making this link between like cognition that you might see in humans or other non-human animals, and then single cells or plants or something else that you wouldn't really normally associate with cognition. And so they actually get into some good examples here. The meaning for Escherichia coli, which is E. coli, which is a bacteria. It's chemotaxis is the mechanism that they look at, and maybe potentially gives meaning to things. So like I talked about a chemical gradient, that's basically what this is. The cell will be attracted to certain chemicals or certain parts of a chemical gradient, and so that's they use to build meaning. And then they have this thing called an RF, which is not sure what it is. But they basically talk about these, it could be reference frames, that, they talk about these different things and how they require energy and how they're state space attractors, and get into some Bayesian expectation modeling. So a Bayesian model, only meaningful differences are detectable. So basically, this kind of reminds me, this model of like, a lot of just basic signal detection mechanisms. So like basically cells are detecting signals in the environment, and they're kind of using those as a basis for like a little bit more structure. And then the question is whether it's sort of cognitive or whether it's something else. This is an interesting paper. I think Jesse in particular would be interested in this paper, reading it all the way through.
I'd like to. Are you just gonna go over the first one next Saturday, or are these the ones too?
I might actually talk about some of these next Saturday as well. I don't know, but I just wanted to put them out there for people so that and we can follow up on them, whenever. But I will put the link to the papers in the chat, so people want to check these out further, as usual.
I'll definitely be looking into.
Yeah. So then for something completely different, this is a paper on multi-axial self-organization properties of mouse embryonic stem cells into gastroloids. So the reason I bring this up is because they talk about gastroloids, which are actually a system that we should maybe present on in the group in another week, where they talk about these different little structures called gastroloids. And so these are like little models of embryos that are not like, they have these things called embryoids, which are groups of cells that are sort of grown in culture, and they sort of mimic the beginnings of a neural system. So they can build these neural embryoids, which are like, they take a bunch of embryonic cells that are going to differentiate into neurons. And so what they end up with at the end of their culture are these neurons in sort of like a ball. And so they can examine sort of the basis of these neural systems in a very controlled and sort of simple way. And so I guess this is an attempt to do this with embryos as well. So they're looking at the emergence of multiple spatial axes in embryos. And so the emergence of multiple axes is an essential element in the establishment of the mammalian body plan. This process takes place shortly after the implantation of the embryo within the uterus and relies on the activity of gene regulatory networks that coordinate transcription in space and time. And so they've used genetic approaches to understand this process. So you have like an embryo or some cells in culture, and you analyze the gene expression patterns. So they've revealed important aspects of this process. A mechanistic understanding is hampered by the poor experimental accessibility of early post-implantation stages. So here we show that the small aggregates, what was that?
They're neglecting the physical again, the forces on the embryo, as it would be as if they had left it in the embryo. They've not taken that into account by the looks of it.
Yeah.
That comment.
Yeah. So here we show the small aggregates of mouse embryonic stem cells when stimulated to undergo gastrulation-like events and elongation in vitro, can organize a post-occipital pattern of neural, mesodermal, and endodermal derivatives that mimic embryonic, spatial, and temporal gene expression. So again, they're neglecting certain things about the embryo, but they're kind of taking these out of context. So they're doing the same thing that they're doing with these neural embryoids, where they're just creating sort of a very simple model of the cells as they're differentiating. The establishment of the three major body axes in these gastroloids suggests that the mechanisms involved are interdependent. Specifically, gastroloids display the hallmarks of axial gene regulatory systems, as exemplified by the implementation of collinear Hox transcriptional patterns along an extending anterior-posterior axis, or antero-posterior axis. These results reveal an unanticipated self-organizing capacity for aggregated ESCs and suggest that gastroloids could be used as a complementary system to study early developmental events. So Susan brought up the point that they're not taking physics into account. And I think this is meant to be like sort of a model system, but just a way to look at like gene expression and gene expression changes in space. But of course, if you don't have physical forces, there are things that are associated with physical interactions in cells, even at the level of gene expression, that aren't going to be picked up here. I think we've talked about that in an earlier meeting where we talked about like how cells, depending on the surface that they're plated on or that they exist in, will express genes differentially, especially when they're differentiating. So this is some of this work, kind of walks through some of the things these gastroloids look like. So, they don't really look like embryos, but they try to build this 3D sort of blob and get similar results that they might get out of a real embryo in terms of cell differentiation. So this is, I think, another interesting paper. I'm not really familiar with this model system, but I think it's, just something that, we get exposed to here, just a little bit. And so if you're interested in following up on this, we can as well. Yeah, the forces of the embryoids will be different from what's in the embryo. Do we have any other questions, or are we any comments on any of those papers?
Just I put in the text box there. Do you want the paper that I know of about optical coherence tomography histology? I know it's off topic, but
yeah, you can send it along. Yeah, why don't you send it along, and I'll send out an update, like a follow-up email, and people can get access to it.
I think it might be a nice way to do histology because you don't have to take a biopsy.
Okay.
But it has to be proven, and Krishna's method of checking histology images might work to check to see whether the optical coherence tomography was working. I don't know. It was just a thought.
Yeah, we can see that. Can be an interesting thing. I have a question regarding the Neurocortex paper.
Yeah.
Yeah, it's just a thought that I've read a few papers regarding the Alzheimer's disease, where the distance between the synapses increases, that leads to random memory loss. Can we can we compare it to the normal brain and get some insights into the neurocortex? Like you see that if the things are distant, that thing happens, and it's just a random naive thought. But if we take certain brain diseases and compare the MRI or whatever it's the topology of the brain at that time, and compare it within a healthy brain and see that how things goes. Like some people who are dyslexic, or some people who don't have they have below the IQ of 70, who don't have much of an intelligence, and we can compare the results and see that what is the part of the brain, or you can say what is the topology, what is the real thing that helps us to summarize the meaning. We can see the brain, even though work has been done on that, we can see the brain of creative people and compare it with the normal people and the below normal person of creativity.
Yeah, they do this in MRI. They actually look at the structures. So they'll compare like Alzheimer's versus a normal brain, and in Alzheimer's are different markers, like the brain will atrophy, so the tissue will shrink, and then you can detect this using MRI, fMRI, to see where activity is. So people have done, built things called brain networks in fMRI. So they build, they'll take like an fMRI signal, which shows sort of, blood flow activity at different points in the brain, and you can segment the brain in different ways. And you can get they have what they call voxels, which are the units of measurement or larger regions, and then you can compare regions between them and build these networks. And so people actually have built brain networks like this, where they have they're comparing different regions of the brain or voxels, and then they can actually make those comparisons of topology. So those papers, I think, probably exist. If you want to read more about it, I would look up maybe
the
brain networks.
Organization, that's also under the INCF. virtual brain that is also under the INCF. They are working on something like that.
Oh, okay.
Nice.
Yeah. I think there's going to be it'll be topology, but it'll also be network activity. So you'll have a topology. You might have changes in a topology, like in an Alzheimer's patient where they might not use parts of their brain, or they use them differently, or but then you also have like the activity. So there might be less activity between two regions, but there might still be a connection, and so it would be an interesting. I don't know. There might be some literature on it. It might be like something that's emerging, but that's an interesting point. Yeah. Okay. Any other questions or comments?
Nothing from my side.
Okay. Well, thank you everyone for attending. Thank you for participation. Shruti, nice seeing you here. You're welcome to join our meetings going forward if you wish.
Sure.
Oh, okay. And if you want to follow up on these papers, let me know. Susan's going to be sending me some things on optical coherence tomography. If you have any comments on the papers, you can put them in the Slack channel or whatever, and we'll maybe talk about it next week as well. So have a good week. See you on Slack.
Bye.
Yeah. All right. Bye.